Okay, so it's a real privilege to be here today to share this day, and thanks very much for the invitation to do so. So I'd like to start with some of my own personal reminiscences of being with Boris, and although I'll have my own little different take on it, I still think you're going to hear a lot of commonality, and we've had a lot of shared experiences. So like Ed, I was a 2T with him. I was a Harvard undergraduate, came to his lab in 1977. And my own situation had some similarities but one difference. So I was a kid from New Jersey who'd never really been anywhere. And I landed at Harvard in 1977. But what I did have was a whole bunch of advanced placement credits. So I entered as a second-year student at Harvard, which meant that I had to choose a major right away. And I liked biology. I had a good high school biology teacher. He actually worked a little bit of Lasko stuff into the AP biology course that he taught. And so I chose biochemistry as a major, got a phone call and said, okay, you've been assigned to a tutor, and it's Professor Magasanik at MIT. Okay, so I had to figure out how to get to MIT. So I started there. There was my dorm. And then I went out into Harvard Square, like that at the time, a little bit pixelated, and made my way through Central Square. There used to be a lot of Scientologists there. And the Necco plant was still the Necco plant at that point. And then I got down here, and And then I had to find my way to Boris's lab, and I think that arrow points directly to his lab on the 4th floor of Building 56. So, and that's what this area looked like when I think that picture's from 1975, so just before when I came. So by the time I got here, I was thinking that this low social caste thing was kind of working against me at Harvard, and I'd been, somehow exiled. But I rapidly. But it didn't take me too long to figure out that this was actually the luckiest break I could have possibly gotten. And many of you, those of you who've had this experience, the tutorials he gave were just unbelievable. And so when I was there, a big book on the operon had just been published by Cold Spring Harbor Press. And we just went through these reviews in the operon book. I used to be able to imitate him saying the operon book. And he just had a beautiful way of doing it. At first he was this very distinguished man who was willing to spend an hour with an 18-year-old kid taking me through this material. And several of you have said that what he did is he taught you how to think. It wasn't about memorization, it was about real understanding. But he also had a way of, he was incredibly rigorous, but he also had a way of doing that never got into intimidation. If you weren't getting it, he was patient. Just keep doing it until you got it. So it was a wonderful experience. He made science fun. He did his science with elegance, and after that, I knew what I wanted to do for my life. The other thing, he was also a very generous man, and several of you have alluded to that. And so even that first year, he offered me a position to work in his lab that summer. And my parents wanted to be back at home that summer, so when I said no, I couldn't do that, he sorted me out a summer position at the Waxman Institute, which was near where I lived. And then the following year, I went back into his lab in September, and then spent the following year working with him, and ended up doing an undergraduate honors project in his lab. So once I got to work in his lab, then I had another really memorable experience from Boris, which was Boris's lab meetings. And they were really, Boris's lab meetings. And they were really quite an experience. So one of you, I think, has already alluded to this. So if Boris fell asleep in talks, in lab meeting, it was all the time. Because he knew, when you're there summarizing your project for everybody, he knew all about it anyway, right? So he didn't really need to hear that. So he would often assume that position. And we had, at the time, the lab meetings were in a seminar room that had a great big table. So we all sat sort of like this one in the picture here. And so there was another aspect of this which involved Boris's shoes. So he would be in this position, and at the time he wore loafers or slippers or something, and he would either kick them off or they get loose on his feet. So it became very fashionable if you were sitting near Boris to see if you could kick his shoe a little farther under the table, out of range of his leg. and when he would stir at a certain moment, he would get this beautiful quizzical look on his face. He didn't really want to show that he was looking for his shoe. So this kept us amused. But the other really memorable part of this is, and this happened very frequently, is that Boris would be sitting like this, and then suddenly you got from him the question you couldn't answer. So you thought you were home clear, he was gone to the world, but he would suddenly, stir and either provide you with an insight about your project that you hadn't thought of, or just ask you something that stumped you. So it was pretty unsettling for me to realize that even with maybe 10% of Boris's neurons firing, he knew a lot more about what I was working on than I did, and I'll never forget those experiences. So out of all of that, I got my first paper published from that undergraduate project, and this is another example of Boris's generosity, because he's not an author on this paper. And so the senior author on this paper was Marjorie Brandress, who was a postdoc I was working directly. And Margie was on the job market at the time, and they reached a decision. I guess we've also learned that Boris didn't like being middle author on things. But they reached the decision that it would be better for her job prospects if she had a senior author publication, and so he's just acknowledged here on the bottom as are you, Aaron. And she got a job at UMD&J just after that paper came out, and I think she's still at UMD&J, so she's done very well. So what happened to me after Boris's lab? Well, I stayed a scientist, and I decided I liked MIT more than Harvard, so I did my PhD with Mary Lou, who's nice to see her here in the audience today. And in Mary Lou's lab, I did a lot of fly genetics and did a kind of saturation genetics screen on a particular chromosomal region in her lab, which came out in genetics. And I also worked on a muscle protein, spent a lot of time trying to coordinate which mutant affected the muscle protein gene. That was not an easy thing to do in the mid-1980s. In fact, I never managed it, but we did work on the muscle protein. And that protein actually has become of interest recently, I've just learned. It's involved in it's a regulator of myoblast fusion, so there are other groups that are working on that now. After that, I went to Cambridge, England, and started working on a very similar project to the one I worked on in Mary Lou's lab, but this was now about the time in the Drosophila world where developmental genetics was really starting to take off with Janni Nusslein-Volhard and Eric Wieschaus and Trudy Schupbach. And I got involved in this particular developmental process. So this is an old picture from when Boris was a child. And it shows that at the posterior end of the Drosophila embryo, or this is actually the oocyte before fertilization, there's a different region of, a morphologically distinct region of cytoplasm that contains these little particles called polar granules. Now in the fly embryo, after the egg is fertilized, the initial nuclear divisions are not coupled by cell division. So the nuclei keep migrating, and they migrate out to the periphery of the embryo. And the ones that migrate into that polplasm behave differently than all the other ones, in that these nuclei cellularized first, and those are the primordial germ cells, which we, for some reason, like to call pol cells in the fly. So what that means is that there's a cytoplasm, the polar granules are cytoplasmic determinants of cell fate, which means it's not, at least initially, transcriptional regulation, rather it's RNA-based regulation, or could potentially be protein-based regulation. And so these RNA-based genetic regulatory processes have pretty much occupied me for the remainder of the subsequent lots of years of my scientific career. So out of these famous screens that were done, this one was actually done by Trudy Schupbach and Eric Wieschaus, a set of genes were identified that gave this phenotype, and these are maternal effect genes. So these are genes that are expressed during oogenesis in mom, and then all embryos or larvae produced by that mom then have that phenotype regardless of dad's genotype. And so here these are the kinds of embryos that are produced by females that are homozygous for a mutant in this gene called Vasa, and they basically don't make the posterior part of their body plan, and if this were a different staining, you could also see that they lack those pole cells that I told you about. So while I was in Cambridge, we molecularly characterized the Vasa gene, and we found that it was related to a translation initiation factor called EIF4A. We know now it's not the EIF4A homolog, but it's part of a large protein family of RNA binding proteins, and that got me launched into RNA-based regulation. This paper, that was the third one of these related genes that was identified. There are many dozens of these genes in every genome. They're conserved from bacteria to people, and so we were able to co-publish here on a little scientific correspondence paper that delineated the protein family. And it turned out, we showed the next year, that this Vasa protein actually is a component of the specialized cytoplasm that's back there at the posterior end. This is a little bit different picture. This is done while the egg is forming. Back in those days, for technical reasons, it was actually quite difficult to stain the later stage egg. We've now solved that with GFP and things like that. So I worked on Vasa for many years, and just show you a couple of the things that came out later. So we had a series of papers looking more directly at how Vasa regulates translation, and I guess the key one was this one, where we were able to show that Vasa interacts both functionally and molecularly with something that we called this at the time, but that's turned out to be another translation factor, eukaryotic initiation factor 5B. And so that's led to a model of how Vasa interacts with RNA and regulates translation. So you can imagine a target RNA in a repressed state. EIF5B is involved in recruiting the large ribosomal subunit, so it's the final step of translation initiation. There is ample evidence, actually mostly in yeast in fact, for translational repression at this step with the 40S ribosomal subunit poised at the start codon. And so what we imagine here is that Vasa can, and we don't just imagine it, we actually have some evidence that supports it. The idea here is that Vasa binds to a target RNA and thus preferentially recruits EIF5B to that target RNA, and thus the 60S ribosomal subunit, so the translation can proceed. We haven't yet quit with Vasa yet. We're still learning things from it. So this is not my work. This is work from Bill Therkoff's lab at UMass Med. And they published a really lovely Cell paper a couple of years ago now, showing that Vasa is also involved in piRNA biosynthesis. And so basically what's going on here is that there's a nuclear complex of piRNA precursors. That nuclear complex involves another one of these dead box helicases called UAP56. And then there's a process. Vasa in early oogenesis accumulates just outside the nuclear membrane of the germline cells. And so their model is that the UAP56 and Vasa are interacting and probably using helicase activities to pass these piRNA precursors out from the nucleus to the cytoplasm, where they're then further processed in the cytoplasm and into the mature piRNAs. So we've started a collaboration with Bill's lab to try to understand more mechanistically how all this is working. Haven't just worked on Vasa, we've done a few other things too, but they've all been relating to, mostly been relating to translational control. So another translational repressor protein that we looked at, we found a new mechanism for how these translational repressors can operate, and basically what this does is it recruits a protein that's related to the cap binding protein that normally catalyzes recruitment of an mRNA, but it's a cap binding protein that can't bind the other initiation factor that's required to get that indirectly, that through another initiation factor binds to the ribosome. So it basically ties up the RNA in an inactive bundle circle. And I guess that's shown here. So this is the translational regulator binding to the 3' UTR of the target RNA. This binds this cap binding protein, which is unlike EIF4E. This one can't bind EIF4G, so this prevents a cap binding assembly from forming on this messenger RNA. And we've worked on another one. And this, again, it's a different mechanism. This translational repressor works by promoting deadenylation of its target RNA. All of these ones that we work on are involved in establishing embryonic patterns. So I showed you the embryos earlier in the talk that lack posterior. This is the reverse. These embryos have a mirror image posterior, so these are 3 posterior segments, and then it's repeated again in the front, so they lack a head and they lack a thorax. And this translational repressor is involved normally in repressing posterior RNAs at the anterior end. So when you don't have it working properly, then you get misexpression of those RNAs at the anterior end, and you get an aberrant body plan. Okay, so getting back to Boris, what are the lessons that I've tried to remember from him to guide my own career? One very important one is the importance of teaching. And I think the MIT biology department is really a paradigm of a place where people do absolutely outstanding research and are absolutely outstanding teachers. It's just incredible here. And I think we're suffering a little bit in a lot of other places that are putting researchers in research institutes, and teachers in biology departments are doing mostly teaching. And what people like Boris and many of you sitting in the room are able to do here is just remarkable, and I think it really remains the model that we should be following for the research enterprise. And the other point I've tried to remember from Boris is that the reward of building up a department is worth the pain in the neck of taking on an administrative position. So I was 10 years chair of the biology department at McGill. You guys don't have to worry about us, but like you, we've built a department from something that was at one level, I think, to something that's at a quite a bit higher level through the recruitment of mostly the recruitment of junior faculty. And there's an awful lot of reward for identifying the right people and getting them to come and mentoring their early career and watching them build a life and a home and a research career and a teaching career in the department. So with that, I just want to say thank you to Boris for getting me started and giving me the chance, and thanks to all of you for inviting me here and listening.